Organellar genome, mitochondrial DNA and chloroplast DNA. History of organellar genome. 1962, Harris and Plot first demonstrated presence of DNA in chloroplast. 1963, Nass and co-worker proved the existence of DNA in mitochondria. Important features of organellar DNA: replicates both in chloroplast and mitochondria in a semi-conservative fashion. Liable to mutation. They are expressed and inherited separately from nuclear gene. Are all aerobic animal and plant cells. They are transcribed and translated within the organelles. Uniparental inheritance is observed. Present in multiple copies in each organelle. cpDNA 20 to 40 copies per chloroplast, 4 copies per mitochondria. In yeast, eukaryotes, plants, cyanobacteria, proteobacteria, archaebacteria, ancient proteobacterium. Then origin of mitochondria, early first cyanobacteria, proteobacteria and archaebacteria. The host that acquired mitochondria, ancient proteobacterium, origin of mitochondria, early diversification of eukaryotic lineage and gene transfer to the host, ancient cyanobacterium, ancient protozoans, origin of plastids, early diversification of algal or plant lineages and gene transfer to the host. Why endosymbiotic theory considered true? New mitochondria and plastids are formed only through a process similar to binary fission. Both mitochondria and plastids contain DNA that is similar to that of bacteria, both in their size and they are having a circular form. These organelle ribosomes have like those found in bacteria less than 70 years, and proteins are of organelle origin like those of bacteria use N-formyl methionine as the initiating amino acid. Much of the internal structure and biochemistry of plastids, for instance, the presence of thylakoids and particular chlorophylls. Chlorophylls is very similar to that of cyanobacteria. Mitochondria have several enzymes and transport systems similar to those of bacteria. Difference between organellar DNA and nuclear DNA. Organellar DNA found in chloroplast and mitochondria, usually circular except in ciliate protozoa where it is linear. Then synthesis continues throughout cell cycle. Organellar DNA found in chloroplast and mitochondria, usually circular except in ciliate protozoa where it is linear. Synthesis continues throughout cell cycle. The nuclear DNA found in chromosomes, linear in eukaryotes and circular in prokaryotes. Synthesis occurs only during interphase. The organellar genetic code has slightly drifted from the universal code and slightly ambiguous in nature. Ambiguity in the genetic code in chloroplast and mitochondria. Initiation codon. Codes found in mitochondrial genetic code: methionine initiation, methionine elongation, ter and ser termination region. Mitochondrial genome, mitochondrial DNA characteristics may be circular or linear. Size varies from 16.5 kb to 100 kb. Empty genome shows large variation in size compared to that of chloroplast. In higher plants, empty DNA has short homologous sequences, recombination within which generates smaller subgenomic circles present in the full genome. In most animals, the two strands of empty DNA have different density, so they are called the H-heavy and light strands. Both strands encode genes, although more are on the H strand. Empty DNA codes for rRNAs, tRNAs, and protein molecules, mainly protein subunits for respiration. No structural DNA proteins are associated with empty DNA. Within mitochondria are nucleoid regions, each of which contains several copies of the mitochondrial chromosome. Yeast has between 4 and 5 empty DNA molecules per nucleoid, and each mitochondrion has 10 to 30 nucleoids. A start region called the D-loop displacement is a D triple helix. There are 2 overlapping copies of H strand. The D-loop is the site where most of the replication and transcription is controlled. Genes are tightly packed with almost no non-coding DNA outside of the D-loop. Human mitochondrial genes contain no introns, although introns are found in the mitochondria of other groups. Plants, for instance. Generally, mitochondrial DNA encodes tRNAs, rRNAs. Protein subunits include cytochrome C, biogenesis, polypeptide of cytochrome oxidase, NADH, deoxygenase, ATPase. Then replication of mtDNA, semi-conservative, relies on mitochondrion-specific DNA polymerase. The origin of replication for the H strand is the D loop, and it is initiated by an RNA primer generated from the L strand transcript. After the new H strand is about 2 to 3 complete, L strand origin of replication is uncovered. The L strand origin is on the old H strand. It is uncovered when the old H strand is displaced by the DNA polymerase synthesizing the new H strand. The L strand origin folds into a loop structure which acts as a primer, and replication of the L strand begins. It involves no proofreading, and DNA replication occurs through the cell cycle. Diversity of mitochondrial genome. Chloroplast genome. Chloroplast genome. Characteristics of chloroplast DNA. Double-stranded. Chloroplast contain naked circular DNA, circular, supercoiled, and generally not associated with any proteins. Size varies. Larger than mitochondrial genome. Each chloroplast contains 10 to 16 cm, sometimes 100 copies of its genome. cpDNA contains mostly inverted repeats. In higher plants, cpDNA 10 to 24 kb long, 2 identical copies per genome as an inverted repeat. Machinery for cpDNA replication provided by nuclear genes. Many chloroplasts contain introns, which form two classes: introns of tRNA located on anticodon loop, introns present on protein encoding genes. GC content in cpDNA differs greatly from that of the nuclear and mitochondrial cpDNA. Nuclear mitochondrial DNA, it allows cpDNA to be isolated by CSCl equilibrium density gradient centrifugation. The number of copies of cpDNA varies from species to species. In all cases, there are multiple copies per chloroplast, and these copies are found in nucleotide regions that are also present in multiple copies. The mechanism of cpDNA replication are unknown. Chloroplast ribosomes are distinct from mitochondrial ribosomes. Translation proceeds in a manner similar to prokaryotes. The chloroplast genome contains genes for chloroplasts, rRNAs, tRNAs, some ribosomal proteins, DNA polymerase subunits, translation factors, proteins involved in photosynthesis. An overview of gene coordination between the nucleus and intracellular organelles. Diagram depicts communication between the nucleus, chloroplast, and mitochondria. Environmental signals into nucleus, chloroplast, mitochondria, and closed-talk. Chloroplast, retrograde signaling, anti-retrograde signaling. Organellar inheritance. Organellar inheritance are two cytoplasmic inheritance concerned with either chloroplast or mitochondrial traits that are usually associated with their DNAs. Difference between organellar inheritance and Mendelian inheritance. Mendelian inheritance governed by nuclear genes exhibits distinct segregation pattern. Reciprocal differences are not observed. It does not show maternal effects. Genes are easily mapped. Nuclear genes are associated with the chromosomes, but organellar inheritance governed by organellar genes does not exhibit distinct segregation pattern. Reciprocal differences are observed, show maternal effects. Mapping is difficult. Plasma genes are associated with either cDNA or mtDNA. Important features of organellar inheritance. Reciprocal differences. As organellar genome from only one parent, generally the female parent, are transmitted, uniparallel inheritance will be taking place. Important features of organellar inheritance: lack of segregation, irregular segregation in biparental inheritance, somatic segregation, association with organellar DNA, mutagenesis, lack of association with the parasite, symbiont or virus. And significance in plant breeding. Role cytoplasmic organelles in transmission of characters. Role in mapping of chloroplast and mitochondrial genome. Role of cytoplasmic male sterility. Mutation of chloroplast DNA and mitochondrial DNA leads to segregation of new variants. mtDNA is used for the study of human evolution.